But it's going to be about the molecular genetics of uveal melanoma, of course, and the impact on prognosis. I'm from the Netherlands, from Rotterdam actually. The Netherlands is a very small country, very close to England or UK. And Rotterdam is about here. This is where I work, the Erasmus Medical Center. We're into, it's a building place at the moment because we're getting a new hospital. It will be finishing up in 2 or 3 years, and next year our new department is going to start. It's going to be very nice, that's what they say us. In Rotterdam we have the Center for Expertise for Uveal Melanoma, and it's actually a collaboration between the Rotterdam Eye Hospital and the Erasmus University, the clinical genetics department, the pathology department, the medical oncology, the radiotherapy. So we all work together within this group so we can talk about all the patients and also about the new research strategies. You all know that ocular melanoma is the most common form of intraocular tumor, but it's very rare. And it's only 5% of the total of melanomas, so there isn't very much attention for it. We have still an instance about 7 per million per year that get a tumor, and in the Scandinavian countries it's a little bit higher, in the Southern European countries it's a little bit lower, but it's approximately about 7 per million. So in the Netherlands we have about 120 to 150 new patients per year, so that isn't very much. It's in the iris, ciliary body, or in the choroid, and the choroidal tumors are actually the most common because it's like 80% of all the tumors. If we look at the disease-free survival for the past 25 years, it hasn't changed a bit. And that's a pity because if you look at all the treatments, because before it was only surgery, you just removed the eye, and radiotherapy came up. And for the patients with radiotherapy, the survival gets a little bit better because those are the patients with the smaller tumors. The larger tumors are now enucleated, so that's when they say the survival is getting worse. But in between, if you look at the group altogether, it's still about 80%. So we have different treatment options, so it doesn't matter which kind of treatment you have for the survival of your tumor. Actually, in the Netherlands we have the stereotactic radiotherapy, that's what we do in Rotterdam. It's like the small to medium-sized tumors and somewhat larger tumors. The brachytherapy is performed in Leiden in the Netherlands for the smaller tumors. And we're going to get the proton beam treatment, and that's going to start somewhere in 2018. We also do the enucleations for the advanced or the larger tumors of the patients with extraocular extension, or if there is secondary enucleation which is necessary when the patient gets complications from the other local treatments. Sometimes we do photodynamic therapy if it's a very small tumor or a suspect nevus. Since a few years I started doing the endoresections in combination with the radiotherapy, and those results are actually very good. And exoresection is also an option, but we don't have that in the Netherlands yet. We have no one to do it. We can take biopsies, but for England it's a little bit different because people are eager here to know what the prognosis is. And in the Netherlands it's a little bit less because patients are reluctant to have a biopsy and they actually don't want to know the prognosis. It's technically possible, but it still has some small chance of complications. You can take a biopsy transvitreal or transcleral, that actually you go just through the eye and at the location of the tumor. That's the easiest kind of biopsy. If the tumor is very much in the back of the eye, you can't reach it because your eyelids are around here. Then you have to do it transvitreal. Let's go through here to the pars plana and just go right into the tumor then. And you can do it also with a needle, or nowadays people are using like a vitreous cutter, just do like a vitrectomy, remove some of the, and most people don't remove the vitreous fluid, just go in, take some tissue and go out. And there's also a biopsy forceps available, which just gets a small part of the tumor. And you get enough material for genomic testing, so that's the most important thing, because you get just like a little bit of tissue and you can test and do actually all kinds of tests on it. You can look, these are actually clinical prognostic factors, but you can also look at histopathology. I will just skip this one. If we look at all those prognostic factors, all will divide into saying that the patient has a good prognosis or a bad prognosis if you've got an aggressive tumor or not. But they're not all conclusive, because you see the spindle cell type, epithelial cell type, and still you see that patients can die due to the metastatic tumor. The same is for the larger versus a smaller tumor. If you have an involvement of your ciliary body or not, also for the vascular patterns. Even if you don't have any vascular patterns, there is a chance of metastatic disease. So all those ancient markers from the histopathology are good, but not conclusive enough. So if you look nowadays at the genetic markers, they're much better in saying what the prognosis is for a patient. And the most important genetic markers, or chromosomal markers, are the losses and gains of certain parts of chromosome. This is a karyogram, so if a cell divides, then you will get 2 pairs of chromosomes. Everyone has 2 pairs of chromosomes. They have actually 23 pairs in the X and the Y. If we look at an uveal melanoma, this is of a uveal melanoma, then we see that we have a loss of a whole chromosome 3. We see a gain of a 6p arm, like we see a double P, they stick together. We see gain of a whole chromosome 8. And that's kind of typical for a uveal melanoma. We can do some techniques. These are the older techniques, like we call them FISH, fluorescent in situ hybridization. And we see, we just color the centromeric part, like the middle part where they're attached. And we see like 4 copies for chromosome 8 then. And we see then here 2 copies, or actually 1 copy for chromosome 3. This actually 1 copy. But if we look at the prognosis, if we divide them based on the chromosome, monosomy of chromosome 3, that's what we generally do, we still see that the disomy 3 patients can develop metastatic disease, while the monosomy 3 patients definitely develop metastatic disease. And then still, this is still questionable because I'm going to come up later on. So if we look at survival, because all those chromosomal tests and things, we do them just to predict the survival. There is actually a new model, because we have some mutations which are available. And this is the new model. I will come to it later on, because a few years ago there was a mutation in the gene described, and it was the GNAQ gene. And a few years later, there was also a gene called GNA11, which are members of the same family and play a role in a pathway which gives actually can inhibit or can where the tumor can grow and proliferate. And GNAQ and GNA11 work here. So it goes through a pathway, and if this gene is mutated, then you don't get this transition and this pathway stays activated. So your tumor will grow and proliferate. So that was a reason to have inhibitors in these pathways, just trying to stop the tumor. Now, if we look at this pathway, there's a lot of genes, and we want to know if this activated pathway plays a role in the development of metastatic disease. We see that it doesn't play a role, because it plays a role in the initiation of the tumor, of development of the tumor, but it doesn't play a role in giving the metastatic disease. So then another gene was described, and it was called the BAP1. And it was described as that the BAP1 was present, or mutation in BAP1 was present in a lot of metastasizing uveal melanomas. So they said approximately 40% of the UMs that metastasized had a BAP1 mutation. And it's located on chromosome 3p21, so that makes it very interesting, because it's also the chromosome which is lost in metastasizing UM. And what is BAP1 actually? It's like what we call a tumor suppressor gene. So you lose one, the other one is mutated, and the tumor can grow. It's definitely involved in cancer development because it's in a complex with a BRCA1 gene. And the BRCA1 is associated with breast cancer. And it has a role in some biological processes like the regulation of a cell cycle and the growth of cell, in the repair of DNA, and this is like the folding of the gene, of actually folding of your chromatin. So it is in a complex, and this complex can, if this complex doesn't function very well, the cells can, or your cells just can grow. So what I have to tell about BAP1, it's like a very large gene. It has like 17 exons, like the coding parts of the gene. And it's difficult just to sequence the whole gene and to see where the mutations are, because the mutations are throughout the whole gene. So there is an easy test just to see whether there is a mutation, because if there is a mutation, you have an absence of protein. So the easiest way to see whether a patient has a BAP1 mutation is just do a coloring, like immunohistochemical test with a BAP1 antibody. And if you don't see any pink, then it's absent. And it's not 100%, but it's quite good at the moment. Here we have the chromosomal makeup of this patient for chromosome 3. And what we do with all patients, we run a SNP array. And the SNP array gives us information on all chromosomes. And this is explicitly chromosome 3. And we see here that there is a part of a loss. While in this patient, we don't see any strange things. And it's like a normal chromosome 3. Has 2 copies. This is a quite difficult picture. This is actually a picture of a collection of some part of our patients. For some mutation, we have 74 mutations tested here, of patients' tumors tested here. These are all tumors with 1 copy of chromosome 3, so depicted here. These are all tumors with 2 copies, and this is here also a part with 1 copy. These are the ones with the mutations, because this dark orange is mutated, and then the inner ring gives us an idea on the expression. So we see that if the patient has a mutation, in most of the cases there's a loss of expression. But even if there is no mutation, like here, you can have a loss of expression. So in most patients it's correct, like you have a mutation, a loss of expression. But in some patients it doesn't work that way. So something else is going on there. If we look at BAP1 and the relation with the survival, or actually with the development of metastatic disease, you see very well that if you have a BAP1 mutated tumor, then the patient will definitely develop metastatic disease. So what's important for this group is that you screen patients earlier. And you screen them so you can find the metastatic disease earlier while they have still a smaller tumor, a smaller liver lesion, which might be treatable. So this is like the same picture again, and we know that gene 11, we see that 90% of uveal melanoma has no role in a metastatic disease, but gives us an idea to verify that it is a uveal melanoma. BAP1 is approximately 50%, and in a disomy 3 tumors, there is a new gene described, and that is the SF3B1. Because if we look, there's a specific mutation in SF3B1 in the disomy 3 tumors, and also in disomy 3 tumors, IFAX was described as also in small percentage. SF3B1 is important because it's like, it gives, I will skip it, but it's a gene which binds to some sequences and can, it plays a role in elongating of some parts of the genes. So it's not very important, but it's important for this Because if we look at our disomy 3 tumors, and we look which patients have an SF3B1 mutation, we see that these patients with an SF3B1 mutation still develop metastatic disease, only in a later stage than when the patients have a BAP1 mutation. So that was actually, at that time, contradicting to the findings of other groups, because they said SF3B1 is in disomy 3 tumors, so it's a good factor if you have a mutation in it. And if we looked at our data, because we have a very long follow-up, we saw them that they got metastatic disease after 7 years. So this is what the model we have at this moment, and it's published last year. If you have no mutation, you're fine. If you have an IFAX mutation, that's actually fine too. If you have an SF3B1 mutation, you still should be monitored for early metastatic disease. It's a little bit later than with the BAP1 mutation, but it's still there. The mechanism might be different, but monitoring is still important, even for the disomy 3 tumors. So what we wanted to do, because just screening for mutations takes some time. There are a few fast tests, but we wanted to know if the chromosomal profile of the patient, like chromosomes, because we always do this SNP array, says something about the specific mutation in the tumor. And we have used throughout the year different SNP arrays, and we just looked at which parts SNPs were on all the arrays, and we did some clustering. And this is like a very difficult picture, but these are all the chromosomes, the 22 and the X, and the red bars are the losses of chromosomal parts, the green are the gains, and then we have some double losses and double neutrals. But it's important that we could divide patients into different groups. And these are the results from these groups, that we have like BAP1 negative but SF3B1 mutated tumors. Tumors which were only without any mutation of IFAC mutations. You don't have to read this, but it was important that we gave the computer the computer just put everything which looked together, look the same together, and then we got the clustering. And if we look specific at the clusters, because if we make a summary out of it, then we see here the BAP1 mutated tumors. We see that they have problems with chromosome 1. We see that they have a lot of loss of chromosome 3. We see they have small problems with chromosome 6, but also with 8. The SF3B1 mutations have a lot of chromosomal aberrations, even on the chromosomes which are generally not involved in uveal melanoma. So this gives us an idea that these tumors are both very different and that they follow a different pathway in the metastatic disease. IFAX mutated tumors are relatively normal, but can still have chromosomal 6 aberrations, but also chromosomal 8 aberrations. And if we look at the major clusters, the copy number variations is what we call these, are placed on chromosomal 1p, 3, 6, and 8, something we knew from many years ago. So if we look at the patient and just look at the chromosomes, we can predict which gene is mutated. Now, coming to the last part, because we can see which gene is mutated. If we can see which gene is mutated, we know that in the Netherlands, because we don't do on a regular basis the biopsies, and we have a lot of patients who are treated by stereotactic radiotherapy, we have a part of a tumor, a part of patients, for which we can't say what the prognosis is. And that's why we think there is a necessity for a non-invasive biomarker, that we can find it in a different way, of predicting a different way. And what we did some years ago, we started out with it, to look at cell-free DNA. And it's like DNA, it is from a tumor, but it is somewhere in the plasma. And we did a small test where we looked at a few tumors, and we had plasma specimens from patients, and we already knew what the genomic variation was. This actually this is a stereotactically treated patient, so we don't have the tumor. And what we did is, these are like the first test. We see that we have here a chromosome 3, and it is lost in the tumor, but it's also lost in the plasma, because the green dots are from the tumor and the red dots are from the plasma. And we also see it from here. Chromosome 6 didn't work out very well, but if we look at chromosome 8, you'll see the green dots are from the tumor and the red dots are from the plasma. We see like that it's going up here. That's an 8q gain. So we see an increase there in the number. So it's a little bit difficult just to get every part of the cell-free DNA out of the blood, but it is possible. So you can look at specific chromosomes in the blood and say and tell the patient what the prognosis is. And because we already know that a combination of chromosomal aberrations predicts the mutation status of the specific mutations, you can extrapolate and just give a prognosis. Another thing we're working on are the microRNAs, and that's on a different level because the microRNAs are small parts of RNA which can inhibit can inhibit some genes. And these are influenceable. So what we did we looked at 26 tumors, and we looked also at healthy choroid, because normally no one looks at healthy choroid, and why does a tumor develop there? And we saw that we had all kind of data on all these patients. If we look at healthy choroid, we did like a PCA plot. This is healthy choroid, this is tumor. So the microRNAs in the healthy tissue is normal, because it's healthy tissue also from other patients, but also from the melanoma patients. We have different microRNAs. And if you look at the 3 different groups we now have, like the SF3B1 group, the IFOX group, and the BAP1 group, then they cluster also in different places. What we now did is look at the BAP1 group, because this is the BAP1 mutated tumors, and we plotted them in a different way, and we identified some microRNAs which discriminate between the BAP1 versus the other ones. And the next step is to look into the in plasma and see whether we can find the microRNAs there. They're a little bit higher, so it's difficult just to pick up the microRNAs we found in the tumor. But what we're currently working on is that microRNAs are packed in little vesicles, and such as those exosomes, and we're now actually isolating these exosomes to see whether the microRNAs are also there. So it's all future work, and it will come up within the next few years. And hopefully, because microRNAs are amenable to therapies, I hope that will be the way just to go for treatment. And actually, everyone working on these projects, there were large parts. There was a lot of PhD students now at the ophthalmology department working together with the clinical genetics, and everyone's working actually only on uveal melanoma. So if you have any questions.
Thank you very much, Emine. If anybody has any questions, could you put your hands up, and Joe and I are going to come and bring you the microphone. So, hands up. Actually, I have a question I can start with. So, am I understanding right what you were saying, that you can actually get some information now from the blood? So even if somebody wasn't offered a biopsy of their primary tumour, you could actually potentially give them some prognostic information from their blood?
Yeah, the test we did at the moment was someone at the moment of when the tumor was still in the eye, which is always draw blood. And now we're trying to see whether we can do it in time. Even if the tumor is gone, if you can see later on in the samples, because we do a follow-up like the first 2 years, every 3 months, and then 6 months, and once a year, where we can see small parts of circulating tumor DNA later on. So, but it is possible, because we have, last week in the Netherlands, it was allowed to have like the NIPT procedure, and that's like the non-invasive prenatal testing. So they're looking at all the chromosomes of the fetus, and it's actually the same technique we're using, only for a tumor. Like a fetus is also some kind of tumor, because also very small. So this should work too. Yeah, in here.
Is it the BAP gene where people are more likely to have metastases? Yeah. What was it? Yeah. I mean, are you following those up? Are those people much more frequently than people with other gene profiles?
Because the follow-up is the same for all the patients, because we don't do biopsies on a regular basis. We don't do biopsy prior to treatment of the tumor. So every patient is followed up every 6 months with ultrasound and liver function tests. And if we see something which is strange, then we will do an MRI or CT and start doing extra testing.
But you have done the profiling on some patients, haven't you?
We have done it
from your research findings. So would you follow up with those more frequently?
Every 6 months should be enough, because a tumor, also a metastasis, will grow very slowly. And if it's visible, then it will go very fast, because you have like a tumor doubling is like exponential.
Thank you.
Hi there. I just wanted to ask you, the information that you're collating about the gene profiling, do you foresee a time where in the future you can feed that back to the drug companies, so there can be targeted therapies for that period where you're waiting for metastases to happen?
No, that is the plan. But the drug companies are at the moment not very interested, because it's just a rare tumor. If you have something like have done the preclinical research and things like that, if you have done that, then they would probably be interested. And it isn't like it's the uveal melanoma on its own, because if you look at the patterns they have somewhat the same patterns, like the BAP1 mutated tumors have patterns that are also in lung tumors. And the SF3B1 mutated patients have patterns are somewhat the same like breast cancer. So it's like a pattern which is also available in another disease, so the drugs used in other diseases might work also for the uveal melanoma.
Do you think that would be the same for those that have metastases as well?
Without metastases?
No, those that do. With this profiling, could that open up other drugs that those with metastases can look at?
That is the planning. We are actually working together with the genetics department. They were testing for the BAP. We're testing our primary tumors for DNA breakage repair. And because the BAP1 is in the cluster with the BRCA1, so they were developing a new test for breast cancer and a new drug for breast cancer. So they were also looking if our BAP1 mutated tumors would respond to it. So that's the way we're working, because it's like a DNA repair problem, which is not, I believe it's not only in melanoma, but also in other solid tumors, and it follows a certain pattern. So we can learn from other tumors too.
Thank you for the talk. It's very interesting stuff that you're doing over there. I'm just wondering, does it make a difference if you're dealing with big or small tumors? Because presumably it's difficult, if there's no mutation involved, just to pick out number of copies in the blood. So if somebody's got a small tumor, would you be able to see aneuploidy on the free DNA?
We don't know that yet, because the genetic testing, normally we have like enucleated eyes, which we do the testing for, and those are the patients we have a lot of samples of, and also the blood samples. So for the stereotactic radiotherapy patients, those are the ones who have like the smaller tumors. And we haven't tested those patients yet. But I believe if you have to work up your material, like you do your blood sample, So you will eventually pick it up if you sequence like deep enough. You have to have enough runs just to pick it up.
I just have one more, if that's okay. How do you get people to agree to giving you bits of their healthy choroid?
No, those are patients which had a tumor, like a tear gland tumor, so they had to have an accentuation. That's where we get some of the choroid from. But also patients with melanoma, we get one part, we get a tumor, and from the other parts, we get like healthy choroid and see whether that corresponds to each other.
Oh, okay, yeah, that makes sense. I thought you just randomly got people off the street.
Give me some parts now. Those like healthy eyes, but they have to have the eye removed just due to something else.
Fair enough, fair enough. Oh, one more over there.
Hi there. It's just a general question actually about when somebody has had an enucleation and the testing is done. How long do you know is that piece of tissue kept for? I'm just speaking on behalf of my dad, whose his testing went to Liverpool, and we actually weren't. It's another story, but we weren't informed that it went. I kind of found out that it was there, and we found out he was class 2 high risk. But I'm just wondering, in case anything came up in the future, is it worthwhile kind of keeping tabs and tracking where it is? And
I think in most centers, patients which are in a study, the material is kept actually for ages.
Okay, I'm not referring to the study. I'm just referring in general.
In general, I think
that are taken.
there are regulations per country, and I don't know what it exactly is because I get out the material before they can throw it out. But I think it's like 20 to 25 years they have to keep it.
So I work with Professor Coupland in Liverpool. We actually do the genetic testing in Liverpool. The pathology department there, I think the regulations are we have to keep the samples for at least 15 years. Yeah.
Thank you. Great, that's everything then. Thank you very much.
Thank you.
Kilic.